probably get started now.
Yeah, okay.
And if I'm not mistaken, is this our last jam session for so this is the last jam session for the academic year, and what a way to end a great year of sessions from so many great speakers. So first, good afternoon, everyone. As I mentioned before, it's really great to see you all. Just one quick announcement that you may have seen a registration experience survey pop up when you came in, and it will pop up again towards the end of the session. So we would really appreciate it if you complete that. We take the feedback that you all provide about the jam sessions very seriously and make improvements based on the wonderful insights that people provide. So without further ado, I'm really excited and honored to be able to introduce Dr. Clayton Yates. Dr. Yates is an internationally recognized expert in health disparity research, cell biology, molecular biology, and molecular pathology. He is the inaugural John R. Lewis Endowed Professor of Pathology, Oncology, and Urology at the Johns Hopkins University School of Medicine. He was recruited to join the School of Medicine in 2022 and is faculty in the Division of Urologic Pathology in the Department of Pathology, where he serves as the Director for Translational Health Disparities and Global Health Equity Research. Dr. Yates holds joint appointments in the Department of Oncology, where he serves as the program co-leader for cancer genetics and epigenetics, and in the Department of Urology. Prior to joining us here at Hopkins, he was professor of biology in the Department of Materials Science and Engineering and director of the Center for Biomedical Research at Tuskegee University in Alabama. Dr. Yates earned his PhD from the University of Pittsburgh School of Medicine in molecular pathology and a certificate of training in tissue engineering and regenerative medicine from the McGowan Institute of Regenerative Medicine. He then went on to complete a postdoctoral fellowship at Emory University School of Medicine, Department of Urology. And after completing his postdoctoral training, he started his tenure track career as assistant professor at Tuskegee University in the Department of Biology and Center for Cancer Research, where he was then promoted through the ranks to full professor. He maintains adjunct faculty positions at Clark Atlanta University Department of Biology and the Department of Pathology at the University of Alabama. Dr. Yates has many notable accomplishments, but one of the things to make note of is that he was able to he and his lab identified and developed a novel therapeutic that targets a specific immune cell termed tumor-associated macrophages, or TAMs, that contributes to increased tumor aggressiveness and therapeutic resistance. This therapy was recently licensed to Aurinia Pharma in October 2021 for clinical development and is poised to enter clinical trials. So congratulations to you and your colleagues for such a great achievement. He and his lab have been continuously funded by NIH and the Department of Defense for over the last several years, and has trained 15 PhD students, 28 master's level students, and 5 postdoctoral trainees. One of the things that I wanted to make note of is Dr. Cooper explicitly said that he is highly regarded as a wonderful mentor. And so that's a really great characteristic of his career. And 5 of the mentees among his many mentees, 5 have received tenure at their respective universities. In closing, just to highlight some of his other achievements before we get to some of the fun facts about him, he's featured on NCI's website the National Cancer Institute for the National Cancer Institute Annual Plan and Budget Proposal for fiscal year 2023, which was provided to Congress and to President Biden and the public. He has also received numerous research honors and awards, has authored over 80 peer-reviewed publications, and is a member of the editorial board of Scientific Reports. He serves as the co-director for the Transatlantic Prostate Cancer Consortium, which focuses on understanding the tumor biology in Native African men in Nigeria. Yay, my family's Nigerian, so thanks, Dr. Yates, for your great work. And he co-chaired the 2022 American Association for Cancer Research, or the AACR conference in April of 2022. He served as the chair of the Minorities and Cancer Research Council in 2022 to 2023 within the American Association for Cancer Research, which serves over 7,000 minority cancer-focused scientists, and is the principal investigator of the NIMHD research centers at Minority Institutes, a site PI of the CTSA, and a PI of a U54 Cancer Health Disparities grant with Morehouse School of Medicine and the University of Alabama at Birmingham. So he has so many wonderful accomplishments, but we're going to actually try to figure out a couple of fun facts about him. So let me try to give you a clue about Dr. Yates. This is related to water and the ocean specifically. So what is an activity that you think Dr. Yates enjoys doing? Very good.
That's right.
Excellent. Yes. So deep, okay, parasailing. That was really great guess, Dr. Cruz. That was not it, but we commend you. Dr. Yates, would you like to tell everyone what the answer is?
Yeah, deep water diving.
Awesome. And how long have you been doing that?
Oh my goodness, since I was a graduate student, many years, over 20 plus years. Yeah, 20 plus years. Yeah.
Just out of curiosity, what do you like about deep water diving?
It's really serene. You sort of black out, it's just you and the ocean. It's very quiet and it's really beautiful down there. So I've had the advantage of diving in some wonderful places. And it's just another world. So it's like this explorations. I think it's the scientific part of me.
Oh, awesome. That's great. And okay, another fun fact about Dr. Yates. So let's see if you all can guess this. This is a kind of genre of movies. Maybe genre is not the right way to put it, but it's a group of movies that has a lot of different and cool characters. If I get any more specific, I'm going to give it away. Westerns, country westerns, Harry Potter. These are all great guesses. Maybe I should be more specific. There are Marvel. Yes, great job. Yes, thank you, Deneen. Wonderful job. Yes, so you enjoy Marvel movies. So do you care to share more, Dr.?
Yeah, no, actually Star Wars too. So they're equally. I love the Disney Channel. So no, I like both Star Wars. I'm a Star Wars fanatic as well as Marvel comics. I just enjoy watching the superheroes and all the entanglements of all the movies. I think they've done a great job.
Awesome. Awesome. Well, oh, thank you. And Dr. Cruz has an emoji that is appropriate for these things. Yeah, that's a great question. Who is your favorite superhero within the Marvel comic movies?
Oh man, I like Iron Man.
Awesome. Well, without further ado, I'm going to turn it over to Dr. Yates, and thank you so much for joining us this afternoon. We're really looking forward to hearing what you have to share and learning from you and your experiences.
Well, thank you very much, and that was wonderful. In fact, I've never been introduced to theme music before too, so now I'm really hyped, and that was really cool, super cool. So thank you guys. That was really a unique experience. I have a few disclosures. And here they are. As many of this disparities is something that we all have to contend with. And we started to look at the rates, particularly of Black Americans. They continue to have higher incidence across cancer types, as well as increased mortality. And when we look at some of the top cancer types that are driving this, prostate and breast are right at the top of this. However, I will admit that that the other cancers, particularly colorectal and now pancreas cancer, which I'm going to talk a little bit toward the end as well, are some of the mostly emerging disparities as well. So when we start to think about this, and those are typically U.S. rates, this is not just a U.S. problem. So if we look at other areas where Black individuals reside, we see, regardless of incidence, we see worse outcomes. So if you start to look at the difference between, Europe as well as, African countries, you see worse outcomes. So this is not even just a U.S. issue; it's a global factor. And, again, as a molecular biology person, as well as just to appreciate the history, we understand that a number of Americans, because of the transatlantic slave trade, have West African ancestry, right? And so, it's really incumbent of us to sort of try to tease out, and I think it's really paramount to tease out disparities in the U.S. You have to also include Native African individuals because, right, we have this sort of shared genomic background as well as the multiple factors that we know African Americans face in the U.S. And so that leads me into this slide. I always love to present this because we don't work in a vacuum, right? Disparities is a multifaceted issue. We have also race and diversity in research. So we have the way people ask the questions. I think we saw that from the last All of Us data that was just presented or published in Nature. We have the difference between race and ethnicity, which are social constructs, and I want to touch upon that a little bit in our talk as well today. But we also have access, quality of care, geographic studies, and then tumor biology. And so, again, more of a molecular biologist and more pathology, we focus on that area, but we work in concert with these. And I think some of the most exciting data that we are seeing now is where these factors all intersect. And as once thought we were studying different areas, I think we're all really studying the same thing, right? It's just looking at it from a different perspective. So I really always like to start off with, the early studies when, we knew about some of the social determinants of health, and although they've been defined much clearer more recently, but just when we start talking about the history of disparities, biology was sort of on the back burner, right? And so Ike Powell, who's a friend and mentor, I think did one of the landmark studies, and particularly in prostate cancer, where this was before sequencing too, right? He was doing microarray studies, and what he looked at was, he was looking at patients that came into his clinic, and Ike was at Caramos at Wayne State University, and he would always see these differences, and so I had these philosophical conversations with Ike all the time, and so when he did this this study, what he saw was that African Americans had differential gene expression, by the way, but some of the genes that were most influenced or increased were inflammatory type genes. So we talk about IL-6, IL-1 beta, all of these inflammatory type genes were overexpressed in African Americans. This was followed up by Renee Reams at FAMU University, where she saw very similar. There was this cytokines burst of storm that seemed to be associated with African American tumors. And then Stefan Alms at the NCI, who is also a close collaborator, really showed that, corroborated those findings, is that Again, these were unsupervised analysis. People were just early days trying to figure out if there were biological differences. And what we found, particularly in prostate tumors, was these immune inflammatory rated genes rose to the top as some of the most glaring factors or genes that were dose differentially expressed between African American tumors and those of European. So, when I was sort of thinking about this, we were, largely focused on if that's the case, right? If there is a true biological difference, it had to be associated with an ancestry, right? And it was because I started off earlier talking about, the diaspora, right? And we talk about the role of ancestry through the transatlantic slave trade and the African diaspora. We thought it was incumbent of us to also understand what was the etiology of prostate cancer in Native African and compare that to African American men. So in collaboration with Falakio Dedna and Solomon Rotimi, we co-lead the Prostate Cancer Transatlantic Consortium, which is dedicated to just that. So utilizing our network within particularly Nigeria, but we're now spanning throughout the entire West Coast. Stefan and I wrote this grant. We got a big DoD impact award where we wanted to understand, was this inflammation that people were seeing at the genetic level associated with systemic inflammation? So we did something called serum analysis here. It's an O-link study. So we're looking at protein expression in the serum of Native African, that means Ghanaian men, African American, as well as European. And Rick Kittles, who is what I like to call the godfather of ancestry, ran all our ancestry analysis so that we could also be able to understand the contribution of genetic ancestry as well as these inflammatory markers. And what we saw was consistently that African ancestry, particularly Native African ancestry, Ghanaian patients, had this suppressed immunity signature. So that means they had all of these cytokines and these markers. It wasn't necessarily an inflammatory signature, but it was a suppressed immunity signature. And we could see that in higher, the most high in Native Africans, African Americans, and then Europeans. And if we look at the markers that were associated, that it was like these chemokines, CXL5, CXL1, and everything. So remember those, because I will talk about those a little bit later, what that means. And if we look at what that means to outcomes, so this was the first study we published in Nature Communications about a couple of years ago, where we were able to say that these signatures were associated with outcome, right? And in men of African ancestry. So we look at this tumor immunity score associated with a worse outcome, as well as a worse outcome, particularly for cancer patients. And then we look at the drivers of this score. It seemed to be this sort of immune related, this TNF alpha. These are all innate hyperimmune related genes that were associated with worse outcome in men of African ancestry, regardless of whether it was Native African or African American. So next, we wanted to understand if that really held true in the actual tumors, and particularly at the genomic level, right? So again, we were looking more at the gene. So now we see protein that seems to be systemic. Was there actually any mutational patterns that were different that could be the reason why we see these differences in these inflammatory markers? And so we sequenced the first Nigerian prostate cancer genome, where we performed whole exome sequencing through our consortium here. And what we saw was that, one, it was incredibly hard to analyze this data. One fact that I want to point out before I get into what we sort of found in biology is that, as many of you know, some of the largest diversity is in Africa, right? As far as patients, as far as population of individuals. So when we sequenced this, what we found was that majority of the variants, these VUSs, we call them variants of unknown significance, we could not align that to the reference genome, right? Because the reference genome is usually, European American individuals. So we had a hard time actually aligning our Native African individuals. And so we came up with all of what we call this sort of blue bar here, these variants of unknown significance. And if we started to look at individuals of African ancestry, even African American, and we go back to those variants of unknown significance, we can see that associated with ancestry as well. But while that was really interesting, we wanted to sort of pare down to what we thought was also very important, is that we were able to look at something that we think was more, pathologically relevant, was BRCA, such as BRCA genes. And interesting enough, we found that there was a Nigerian and African American specific BRCA variant that was not annotated in the typical BRCA variants we test clinically, right? And this is really important because we now know BRCA variants particularly are associated with certain types of therapy, including PARP therapies, PARP inhibitors for prostate cancers. And now here we're seeing that African Americans don't typically have this BRCA variant that we test for. They have this more African ancestry specific BRCA variant that we see more of our African enriched individuals. And if we look at that, this is not a somatic variant. This is actually a germline. So we see almost 100% of our Nigerian individuals have this BRCA variant. And this was really interesting. So we got into this, African-American and, what is an African-American? And we're going to talk about that a little bit later as we get into our gene expression data. But what we saw here, and this was the beginning of our thinking about how do we use Ancestry, only our African Americans who had high African ancestry, that means we're talking about over 70% West African ancestry, expressed the BRCA variant. So we talk about our admixed African Americans who had lower than African ancestry, we see that go away. And so that sort of replicates why we see only 68% of the frequency in our African American variants. But if we look at our high African ancestry, is almost 100%, even in African Americans, very similar to our native Africans. Okay. And we don't see that at all in our European. So as we started to understand this contribution, so now we're thinking about this interplay. So we have this inflammatory type genes, we got these mutations. The question is, where do they intersect? And do they intersect and have a role, right? So Israel El-Hussan, who was a postdoc, a graduate student in my lab, and actually moved with me here to Hopkins and now as a postdoc, she wanted to go deeper, right? And so she started performing superpopulation analysis, which I call the Big Five, right? African ancestry, European, East Asian, West admixed population. She wanted to go deeper, right? She wanted to say, is there subpopulation differences? That means East Africa versus West Africa. And can we look at tribal differences as a reason why we see this, maybe this inflammatory genes, particularly in Native Africans? So we looked at our African American individuals, and we ran what I call our superpopulation analysis. And this is the US cohort, and these are all African American men. And then we ran subpopulation analysis. And you can see in this cohort, our individuals were mostly Yoruba Bantu, okay? And so because we had gene expression data, Israel immediately wanted to confirm. And so again, yes, in our African American individuals, we see this high sort of innate immune response that replicated what everyone else was seeing, that African American men present with this chronic type inflammatory type disease. But because we also were able to have genomic information on these same individuals, we ran, our gene expression profiles, and what we found was that, yes, we saw this BRCA variant, but we also saw a really interesting SPOP mutation that is known to be associated with prostate cancer tumorigenesis, but we saw that much higher and higher frequency in our African American than we saw European American. So we thought this was a real opportunity to start to understand the interplay between these genomic variants that we're seeing associated with African ancestry and their gene expression profile. That means how these individuals are presenting in the clinic. So when Israel saw this, right, she was like really excited. She came into my office. She said, well, I think this SPOP is the reason why we see inflammatory type disease. I said, well, if that's the case, we should see that in African Americans, but also African American men who don't have the SPOP individual mutation, right? And so she took our SPOP mutant individuals, and absolutely. So if you look at the gene expression profile, it's this innate inflammatory gene response. I've been saying to you guys, talking about it from the beginning. But if we even look at African American men who don't have the SPOP, and there was only a few because we saw that high frequency, they don't have that classic inflammatory gene that we saw that most people present with, that others have been replicated. So now we're starting to understand the genomic contribution, as well as what people also, there could be reason why African Americans are presenting with this inflammatory genes. Now, we do see other types of inflammation, influenza, HIV, but it's not that classic innate response that others have been seeing that has been replicated over and over again. So what is this SPOP? People ask me all the time, Dr. Yates, you go around talking about SPOP. What is SPOP? All right. SPOP, one, is a prostate cancer gene. We screen for it now genomically in individuals in the clinic every day. And the reason why is because it's a known E3 ligase. So what is an E3 ligase? So we have proteins that are being made, and we got proteins that be degraded. The job of E3 ligases are to chew up. So that means that, a protein gets made, gets degraded, and it stays at steady state. And so it's the equalizer, if you will, in the cell. You can imagine if that equalizer is mutated, right? That means it's not ubiquitinating proteins that are supposed to be degraded and kept at steady state, then they accumulate. and they accumulate and they increase. And so we feel like, particularly in the case of prostate cancer, which is driven by androgen receptor and driven by testosterone, we see very nicely that in the SPOT mutant individuals, androgen receptor, these other immune genes such as PD-L1, they're all accumulated because they're not being degraded because of the mutation. And that's creating a biology that's letting these proteins go haywire in the cell and promote tumorigenesis. So if that's the case, we wanted to look at if these interferon genes were associated with these SPOT substrates. Absolutely, indeed. So we looked at all these interferon genes as innate inflammatory genes. They are associated with African ancestry, and they're associated with SPOT substrates, particularly PD-L1. We looked at PD-L1 expression in our African American cohort or high African American ancestry individuals. It's much higher than our European individuals. And if you look at the co-expression of immune cell types such as CD8, which is a T cell, we also see increased T cell in our high African ancestry than we see in our European. This is really important because prostate tumors have largely not been effective in immunotherapy because they're thought to be immunologically cold. And here we're saying in our African American cohort or high African ancestry, that is not the case. They are clearly immunogenic. And if you overlay this gene expression profile with the mutational status, right? And so I really want to call your attention over to the left slide. We're looking at African ancestry. So these are all of our high African ancestry individuals. These are all of our SPOT mutant. Even if we titrate ancestors, that means we got ancestry individuals between 50 to 80%, and then we got high over 90%. You can even see the gene expression changes with the percent ancestry, even within the SPOT mutant individuals, right? So here we're sort of constructing this interplay between ancestry, mutation, and gene expression, sort of understanding this complex interplay of all of these factors that could be driving tumorigenesis in African Americans. And what does that look like at the tumor level? Indeed, African Americans have hot tumors, right? I talked about before about European men being particularly immunologically cold. That's how immunotherapy has largely failed. These African American tumors are largely expressing these immune cells, again, that are typically absent, and they're co-expressing exhaustion markers, which you would expect, that's suppressing this immunity. Although they're there, it's suppressing this immunity, and we think that's responsible for why we saw that immune suppression score in our serum cohort as well. So we're starting to understand why we saw what we saw in the systemic response. We're seeing that at the molecular genomic level as well in the tumors, even at the single cell level. So what does all that mean, right? So it's a lot of biology, and people say, well, what does that mean, right? Emmanuel Antokarakis, who was formerly at Hopkins, is now at University of Minnesota, did what I think what we're headed to is the future of where we're utilizing this biology, this biological basis of disparity. So what Emmanuel looked at was SPOT mutation status, right? And so the typical progression of a man with prostate cancer is initially diagnosed. That patient's testosterone is depleted, or we call it androgen deprivation therapy. That patient will invariably fail in 18 months. It's a typical central dogma of prostate tumor. And then the second line of therapy will be chemotherapy. And that patient will progress to either castration-resistant and or metastatic disease, and chemotherapy will be the standard of care. But if you think about SPOT, and remember I talked about the biology, PD-L1 we showed in African American, but also androgen receptor was one of that. What Emmanuel said was like, what if I didn't, in our SPOT mutant individuals, and if they fail frontline therapy, what if we don't progress those individuals to chemotherapy? What if we keep them on androgen deprivation? Because if the SPOT is mutated, that AR should not be degraded. It should stay there, and those patients should remain responsible. So that's exactly what he did. And you can see the outcome differences, right? So if you see the individuals with SPOT mutant that did not progress to cytotoxic chemotherapy, they have an overwhelming survival advantage to the standard of care patients that got cytotoxic chemotherapy. This is where we feel like while we're doing the biology of these disparity treatments, and now we're finishing up those studies, and we hope to now over-enroll African American men in the second phase of the clinical trial, right? Because we're matching the biology with the available therapies we have currently. But that also allowed us to propose this larger diagram, right? So we proposed this sort of We published this in Cell Trends a couple years ago. Well, we're starting to understand, right? So I started off my diagram with a proposal of how we thought about doing disparities research. We're now sort of refining that, including some of our genomic findings. When we think about including ancestry, right, we're saying that, you know, and again, there's no secret that molecular sequencing is a part of standard of care in many cancer types. But what we also are promoting is that we need to include ancestry. We need to understand the genomic background, genetic background of these individuals. And we're thinking about buckets of ancestry individuals, high African ancestry, low African ancestry, universal drivers. But we also understand that there's geographical, structural differences, molecular profiling, as well as ancestry and also social determinants of health that have to be incorporated. So we're proposing this more the refined diagram of which we should think about prostate tumors and cancer in general. So in order to do that, we started to set out, and I think, a comprehensive approach. Now, again, forgive us, but we're molecular biologists. And so we also thought about, okay, we had this genomic data, we had this protein data, we had this RNA data. What if we can really start to overlay that in every single individual and start to understand really what's happening? And could we define what should be a therapeutic target in, you know, African Americans? And so we developed this, you know, I think rather large cohort where we're performing this multi-omic analysis. And what we're doing is we've collected a Native African now, African American. We're performing whole exome, which is DNA sequencing, methylation sequencing, as well as whole transcriptome. We perform digital spatial analysis because we need to understand the spatial context within the tumor cells, as well as we've been able to now take a subset of those and do single cells, so we can look at the expression or transcriptome of every single cell within the tumor and understand the cells that are contributing possibly to this inflammatory type response within the tumor microenvironment. So Israel went back again and performed our genomic ancestry. Now we have African American as well as Native African, and so we perform, our superpopulation analysis as well as our subpopulation, and you can see most of our Native African individuals because we've enriched, our Native African now for transcriptomics, and you can see they're mostly Yoruba background. But here's where it starts to get interesting. Okay, this is what I'm really excited about. So we have Native. So at the top of this diagram, we're saying this is self-reported. So we have Nigerian, African American, European. When we utilize our genomic ancestry, and now we think that's around 70%. We got that from our genomic data, our whole exome data. Statistically speaking, if an individual is below 70%, they start to recluster. So we had African Americans, 7 out of our 45 African Americans who had below 70% actually cluster. Their gene expression looks more like European, okay? We had one European who had over 70% African ancestry, but they self-reported as rate. They actually cluster. Their gene expression profile looks like an African American, so they cluster with African American. And if you start to look at what they look like here, right, you can sort of see these are African American, self-reported as African American. These are Yoruba subpopulations. That means, so even in African American, we corrected for ancestry. We still see differences in our African American compared to our Native African, even in ancestry, even corrected for population status, right? So African American men look different than our Native African, right? Even regardless of ancestry. And here's where it got even more interesting is that the individuals whose self-reported race do not match what we call when we perform the genomic ancestry, they look different than everyone else as well. So they don't look like the self-reported African Americans, they or the Native African, they sort of fall in this middle because they're self-reported and the genomic ancestry are not concordant, right? So they sort of fit, they sort of look different. Now, what does that look like to immune score? Again, we see our African American absolutely have the highest immune score. Our Native Africans, although they had tumor suppression, they do not have this inflammatory types response that we saw in our African American, right? So Native Africans don't have it, the immune score or this inflammatory score that we saw in our African American individuals. But what do they do have? So remember, we talked about that serum. We see suppression, right? This tumor suppressor score. regardless of whether it's African American or Native African, it absolutely correlates with African ancestry, while race seems to be associated with inflammatory type response. And then we start to look at that as far as our scores, we see that sort of bear out. And because we, again, wanted to nail down what the actual cell types, we performed deconvolution analysis of our RNA-seqing data, and we were able to start to understand that the cell types responsible for this suppressed score happen to be macrophages. These sort of macrophage myeloid type response genes seem to be associated with this suppressive immune score that we saw in our African ancestry. And that didn't matter if it was a Native African or African American. That M2 macrophage response seems to be systemic. But because, again, this is bulk sequencing and deconvolution, we wanted to know, could we actually see those cells? So we performed another study called multi-omic analysis, where we could look at not only every single cell, this transcriptome, but we could also look at chromatin and remodeling changes at every single cell within the tumor microenvironment. So here we looked at single cells. So each one of these dots represents a cell in the transcriptome. We're able to group that. And so you can, again, we look at these macrophage, we call them myeloid cells. You sort of see this expressed here in our European. You don't see a whole lot of myeloid because, remember, they're supposed to be cold. Immune therapy does not work in these individuals, and we're not expecting a whole lot of immune cells. But when we look at our African American, absolutely. Look how they light up. So these myeloid cells are expressing sort of like this fuchsia color. We see all of these myeloid macrophage cells infiltrate, and they're infiltrated within the tumor microenvironment. So it's not just inflammation. These cells are being recruited, and we think this is possibly why this inflammation is sort of sustained. And if we start to look at the gene expression changes in those myeloid cells, guess what? We're back to that innate response. So, before we didn't know why that innate response. We've now been able to identify the cell types that are responsible for this interferon signature, and it seems to be coming from these myeloid cells recruited to the tumor, sort of persisting and providing the ligands responsible for this chronic inflammation that we and others have been able to see. And because we also had chromatin levels, so that means we actually have structural variants at structural information at the DNA level. What we saw was that not only did we see that these myeloid cells have infiltrated, it seems to be that these myeloid cells are causing structural changes at the chromatin level, again, and causing DNA to be more accessible in African Americans. And these transcription factors located at these regions of open chromatin, these sort of STAT genes, again, all information, seem to be driving that. So you can imagine this loop, right? So you got these ligands, this DNA being open, transcription is going, it's keeping on, and it's sort of driving the tumor genesis of these and why these tumors are more aggressive. Now, when we find these are kind of what I like to call provocative findings, right? But the proof is in the pudding, right? We see the same sort of phenomenon occur in African American women with breast cancer. So if you look at this is a paper, a nice review in Frontiers Oncology. When we were seeing our prostate data, they see absolutely the same thing. Women of African ancestry have this interferon innate response. They have more of this myeloid cell infiltration and suppressed T cell immunity. But what we wanted to do was characterize that further. So we wanted to look at, again, we were classically a prostate cancer lab. What was the contribution of androgen receptor in African American women with breast cancer? And so we looked at the presence or absence of androgen receptor status. And what we found was that in our breast cancer patients, AR was lost. And overwhelmingly 81% of African American women, regardless of subtype, compared to 56% in European individuals. And so I'm not going to go through this because we published a lot of information on that. But what we did was that in our triple negative, we've now defined as a quadruple negative. These are African-American women who present with loss of ER, PR, HER2 status, but also AR. We defined that that's the most aggressive subtype in African-American women. That's associated with a distinct biology at the gene as well as microRNA level. And those women present earlier in the clinic. So they're an African-American woman who has decreased or loss of AR expression presents 6 years earlier than a woman who has presence of AR. And that is sort of mirrors what we know statistics of triple negative in African-American women, but that also holds true for European. They just are clearly diagnosed at a later age. So if we think about those, women, particularly in the age of 48, 40, below 50, those women are presenting as quadruple negative and they have the worst outcomes. We also were able to show that these African-American women, typically these AR negative, present associated with basal subtype. We know basal disease be associated with most aggressive. And if we look at triple negative subtype, again, it fits this immunomodulatory basal subtype. So these African-American women have, just like we showed in our prostate cancer tumor patients, have this immunomodulatory basal subtype. And again, we think this loss of AR is due to diaspora, through the transatlantic slave trade. Sort of the loss of it is acquired. And that's the upregulation of this immune response. And again, we think immunity, or particularly checkpoint therapy, or this whole advent of immune therapy, will be more beneficial in African-American women because of this sort of shared biology that we now see in prostate patients. But I wanted to kind of get back to this, this ancestry as well. We wanted to see, again, what is the contribution of race versus ancestry? So we perform qualified genomic sequencing again, and these are self-reported race, in our self-reported African-American versus self-reported European. We perform qualified genomic ancestry. And again, look how you see this separation, right? You sort of start to see this separatory fashion of these are high African ancestry individuals, these are European now ancestry. And this is really interesting because these are European individuals who have shared genomic ancestry, so phylogenetically cluster with African Americans, and you can see they start to share African American gene expression, right? We went on to follow this up in collaboration with Melissa Davis. We got the cover of Cancer Discovery, where we saw this very, very bad pan out in a Pan-African cohort, where if you can see, African American women from Detroit, and there was no secret that was at Tuskegee from a year. So this is our Alabama cohort. And then we have Native African from Ghana. They share this sort of shared genomic ancestry. But here's also where it gets interesting. These over here on the left, these are Ethiopian. So in the middle, so these are Ethiopian individuals. Ethiopians don't have a predominant West African ancestry. They have shared admixture. So they have half European and half African ancestry. So now we wanted to see if we have an African from East Africa, we have African from West Africa, and we have African American, how does the gene expression look? So if we start to cluster that, right, we have now we have an Ethiopian based signature. We have a shared genomic signature because we know, again, because of the transatlantic slave trade, a shared Ghanaian African-American, that's predominantly here. But then we have genes only expressed in African-American, and then we have some genes that seem to be African and the share between Ethiopia and Ghanaian. Those are not as much. You can see there's not as much share between East and West Africa as we see as African-American and Ghana, but there is clearly a shared group. And if we start to sort of think about what those are, so at the top here, these are what I'm calling African ancestry. These are European ancestry. And at the bottom, we're calling these self-reported race genes. So what are the self-reported race? Oh, excuse me, I'm going the wrong way. So what are the African ancestry genes? Immune related, right? Now, again, where it gets very interesting, if we look at subpopulation analysis, we can see subpopulations share different types of immune response, whether it's East African or West African, although it's still shared African ancestry, right? But it's different if a person is in East Africa or West Africa, right? But if we start looking at race genes doesn't have anything to do with immunology. Race has to do with diabetes, cardiovascular disease, obesity. So we're saying these race genes has to do more with these sort of biological comorbidities. Our ancestry genes have to do more with how we're looking at this sort of, what is basically inherited. Lastly, I wanted to sort of dive in deeper because we think these macrophages are really important. So we published a paper with, we did some work with Kevin Gardner, who is chair of pathology at Columbia University. Our lab for a long time has worked on a marker called Kaizo. We published a lot of papers in it, so I don't have time to go in it. But what we were able to show is, by the fact, it was one of the first health disparities markers recognized by NCI. But what we looked at with Kevin was that if we had Kaizo, a bona fide disparity marker, could we see this immune cell population sort of clustered around it? And we absolutely did. We formed nearest neighbor analysis where we were able to look at high Kaizo patients, and we're looking at immune cell macrophages. And I know this is really complicated, but essentially we're looking at cell types and immune cells that cluster around immune cells that have our classic disparity markers. And what we were able to find is that these immune cells, particularly these macrophages, cluster around these, our high Kaizo expressing tumor cells within the tumor microenvironment compared to European. And we think that's why this immune suppression is occurring, and that also occurred by race. So if you look at what this looks like in mice, so if you take this Kaizo expression gene, again, it's a known disparity marker. We knocked it down in mice. It seems to block tumorigenesis. But not only can we see this sort of decrease in tumorigenesis, it actually reverses this macrophage phenotype. So if I showed you that diagram before, is that African Americans in our prostate have this sort of suppressive macrophages. If you deplete Kaizo, you get more of the good macrophages and not the bad macrophages that cause suppressed immunity. So we absolutely think this is important, not only as a biomarker, but we think it's important in the pathogenesis of disease. And if you look at this in a biological context, that means you take conditioned media from high Kaizo expression cells and you apply it to the macrophages, you can absolutely make those cells become suppressive. But if you take conditioned media from Kaizo low expressing cells, you can actually make them become more of the more favorable macrophages, and they actually start phagocytosing and they start to recognize in cancer cell and participate in immunity instead of blocking it. And so we presented this diagram that not only do we see the suppressed immunity, we actually think there's a biological basis for it, that these Kaizo is causing escape of this immunity. We see Kaizo more expressed in African Americans, and that this is responsible for this immune escape. And in Europeans, we see this high Kaizo, this low Kaizo, and that's also why we think that the Europeans present with this more favorable response, because the immune cells are participating in anti-tumor response instead of suppressing more of immunity. So Kaizo as a transcription factor is very difficult to target. So we wanted to see if we could target what we think was the real culprit, was these macrophages. So in collaboration with Jesse James, in connection with an NCI and not Jelani, because actually Jelani just got an R1 to develop this in prostate tumors, we developed this CD206 inhibitor, which we think is targeted to macrophage, these macrophage or myeloid cell types that we were seeing in both our prostate as well as breast tumors. We were able to show that this macrophage inhibitor not only targets macrophages, it causes repolarization. So sort of like our Kaizo, but we think this is more pharmacologically targetable. So immediately upon utilizing this therapeutic, we can make the bad macrophages become good. So that means they express CD68 and they lose that sort of suppressive response. In mice, we see that's synergized with not only checkpoint therapy such as PD-1 or PD-L1, as well as so eliminating these macrophages is really important. We think in combined therapy, they will be very effective in African American women. And we also think this is important in timing. So with collaboration with Roger Lowe, we saw that a number of tumors, particularly in the melanoma space, and this also holds true for breast cancer patients, when you start to look at, and in this case, they were looking at ERK therapies, or receptor tyrosine kinase, such as EGF receptor therapies, and how these tumors, what the biology of these tumors, when they start to recur, what they invariably saw was that these macrophages enter the microenvironment, right? And so with Roger Lowe, when we published that science paper, he said, hey, Clayton, what happens, if we utilize your macrophage inhibitor, can we actually make our immune therapy more effective? And that's actually the case, right? So if you take tumors at the time they're recurrent, we see that these macrophages sort of populate the tumor microenvironment. And although this was in mouse models, we see this already occurring inherently in African Americans. So we were really excited. So we utilized that therapeutic, and we were able to show that they decrease tumorigenesis and they make immune therapy actually more effective. And because we were developing this therapeutic, we also wanted to know if we can detect these tumors earlier. So we developed an imaging agent in collaboration with scientists at UAB and Candace Parker. And we were able to publish sometime last year that not only can we have a therapeutic, we now have a companion diagnostic where we feel like we can identify these macrophages through radioisotope labeling in humans, and we can identify a recurrence much earlier, and that we can understand when a treatment or when a tumor is starting to recur, particularly in African American individuals, and be able to apply the next therapy. And so we're really excited that we now have a drug, we have a companion diagnostic, and we're moving both of those toward our clinical trials. And again, why does all of this matter? I like to tell a story from a good friend of mine, Oliver Sorted. So Oliver took a lot of this data that we had in prostate tumors, and he applied immune therapy, and he overrode African Americans. And this immune therapy was called Provenge. It's called the PROCEED trial. So Provenge is called, or the biological name is Supacil T. It's an immune therapy, and his rationale was that if because of the biology that many of us were seeing, that they would respond better. And so again, very much like Emmanuel did, as patients sort of progress to that castration-resistant setting, which we know cytotoxic chemotherapy is really not effective, he applied Provenge instead of chemotherapy. And what we say is that African Americans not only responded 2 times better, but they had a 60% increase in outcome compared to European, again, because we matched the biology with the right therapeutic. I like to end there because, I get to, talk about this and showcase this, but the work is really done by many of the trainees over the years in our lab, and they've gone on to have very successful careers. And not only the trainees that are directly in my lab, but the individuals that in our consortium as well. And we think that, this is really important. And not only are we doing the biology that's affecting African Americans, we are also doing the biology that is affecting West Africa, as well as building capacity. And this is just, I'm really thankful to our entire CAPTC group, because they've supported us over the years. And this is definitely a group effort, and we couldn't do it without them. And also thank you to the many collaborators that it takes to participate in over the years and to get this work done. So I'll stop there.
Thank you so much. That was incredible, Dr. Yates. That was just so inspiring. And there are many of us who are watching and listening to you that have a family history of cancer, and we have seen the toll that cancer takes on our loved ones. So to hear about the targeted approaches that you and your colleagues have done is so impressive. And I personally want to thank you not only for the work that you've done and the ways that you've really intentionally sought to build up the capacity of cancer research in West Africa, but also for just presenting in a way that makes this groundbreaking research so accessible. Not all of us are biologists. I myself am not a biologist by any means, but you just did a wonderful job of really showcasing all of the different types of work that you're doing and the breadth of collaborations that you cultivated, as well as just this idea that that we do really need to think about how to center the African diaspora when we're thinking about, strategies that really that get at the genomic origins of health disparities. So just wonderful job. And I see that Doctors Cruz and Cooper have their hands up.
Thank you so much. Thank you.
You're welcome. Dr. Cooper?
Oh, first of all, yeah, I want to apologize for not being able to be here at the beginning of your talk, but also to thank you so much, because I did hear a good amount of it, and was similarly impressed and inspired by your pursuing of questions from one piece to the next piece, and your ability to sort of build those bridges with people doing work that was compatible in order to come up with some really impactful therapeutic and diagnostic modalities. My question is just around to what extent, when you see these differences between African Americans and Africans, to what extent you have looked at sort of the interactions with social and environmental type measures as a way of explaining, some of the inflammatory responses or immune responses you've seen.
Yeah, no, absolutely. So I didn't show it here, but that serum data that we had, excuse me, actually Stefan published in JAMA. So that inflammatory score is correlated with a high neighborhood deprivation index. Right. So if you take that score, and that's kind of how we came up with this race versus ancestry thing too, because we weren't thinking about it like that. We were thinking that Africans were going to share, Native Africans were going to share African American. But when we didn't have the social determinants in our Native African individuals like we had in African American. So when we did that analysis and we only in the same cohort and we only had the African Americans, the inflammatory score shot to the top, and then that overlaid with SES, social determinants. So that is a JAMA paper that we published some just sort of last year. So we're starting to see that inflammation be associated with social determinants, and I think that is the sort of future. And I have a shameless plug, and I'm sorry I didn't include this in our original but if I could share this one slide with you guys, I think, and I'll be happy to sort of we just were funded a Cancer Grand Challenge Award. So this is $25 million, right? To answer that question, Dr. Cooper. So we will recruit 40,000 individuals from the US, UK, because it's a Cancer Grand Challenge, it's a collaboration with the Cancer Research UK, but US Blacks, UK Blacks, East Africa, West Africa, social determinants. Every person would get exposomics. That means we'll measure their environmental impact as far as their blood. Genomic sequencing, every person would get genome sequencing, and then we'll assess their biology of what we're calling the platinum cohort at the bottom. But this will be the largest sort of cohort right now. We're recruiting 40,000 individuals globally.
Awesome.
To address that question.
Very impressive. Well, I'll just put a plug in for the fact that think of us in the Center for Health Equity if you're looking for collaborators in the future, or even in the work that you're doing now.
Absolutely. Absolutely.
Yeah, thank you so much for that question, Dr. Cooper. I was wondering the same thing, and I really appreciate that answer. And congratulations to you and your team, Dr. Yates. That is wonderful. Wonderful. I'm excited to see what you all, the new knowledge.
Well, I think this is us because, and even though, and I apologize, you know how these things are rushed. We're saying that this is going to be it's going to be a publicly available resource. I think it's all of us are going to have to contribute to it. This data will be mine for years. And so this is a Hopkins thing. It's not going to be our lab thing. This is going to be a resource for all of us. And so I don't want to see it as us. I want to think about how we can bridge that gap and add to it, or even increase to colorectal cancer, pancreatic. We are doing pancreatic cancer and other types because we hope to create that framework to be able to ask many questions down the road in the future.
Yeah. That's
so Dr. Cruz still has her hand up. I want to make sure
I'm sorry.
Ask her question.
You can hear me. I apologize. I'm driving, but this is really wonderful, and certainly congratulations on all the work, including the new initiative that you just shared. The same question as Dr. Cooper, so that one's covered. But my question was about when thinking about the application of what in your last few slides you were talking about how you now have both a diagnostic and obviously a targeted treatment as well. And I was wondering how in the care of patients with prostate cancer, how would you envision this would be used? Like would it only be applied to people who self-identify as being African American, or should everyone's tumors be getting the same kind of screening? I'm just curious about how it lines up in a clinical setting.
No, absolutely. So two things, and I'm sorry I went too fast. So first of all, we think, and what we're doing now is, particularly in late stage disease, every person right now, and this is actually a disparity, but NCCN guidelines that our patients should be genomically screened. Okay. According to NCCN. Now, again, I came from Alabama, rural America. It's not happening. Okay. It happens at Hopkins, but it is not happening out in community oncology clinics. So that's one thing, and it's NCI guideline approved as well as reimbursed by insurance. So first of all, we got so we got these markers, these genomic markers that are reimbursable, approved, FDA approved, and should be happening in the community, right? And so that's why I've painstakingly gone through these mutational patterns, because we think those are inclusion criteria. Right, these are ways. So then we talk about the genomic ancestry. So we just initiated a partnership with our preferred sequencing of care since I've been here, and we're going back to all of the Hopkins patients as well as patients we had in a UAB and Piedmont and some of the Piedmont hospitals when I was in Alabama, and we're reforming ancestry analysis with that genomic data from the, again, real world patient data that's used to make treatment decisions every day, and we're understanding this interplay so that going forward in a clinical trial, we can start to say, okay, this patient has this genomic ancestry, they have this gene expression profile, this mutational status that corresponds with this therapeutic. It could be, one therapeutic versus another. And again, this is happening in real world every day, but not for African Americans because the biology is not there to justify it. So this is kind of why we've painstakingly gone through this sort of step-by-step way, and to have inclusion exclusion criteria, because I think if we do just include all African Americans, that's not going to be a recipe for success. And you know how it is. We're only going to get 1 or 2 shots and they're going to say it didn't work. So we got to be very clear how this, when we go into very designed clinical precision treatment modalities, that we define that patient population, and that's why we spent time doing it.
Thank you very much. I just want to make one more comment. There's a lot of parallel here with my favorite condition, which is kidney disease. Would definitely look forward to following up with you about certainly the immune sort of mechanism. I think there's a lot that's very similar in terms of what people are finding about what we understand about disparities in kidney disease as well. So look forward to following up, and thank you again.
Absolutely. Thank you. I look forward to it.
I wanted to invite others to pose questions to Dr. Yates. I see a question in the chat from Kayla Cunningham, who asked, how would patients enroll in your Cancer Challenge grant study?
Oh, that is great. So we actually have several ways, but primarily we contracted with a patient advocacy group. That's actually one of the criteria for the Cancer Challenge calendar. So that's actually led by a native person here in Annapolis called Voices, right? And so she's leading. It is a breast cancer, but we now have patient advocates with prostate. So they will be, primarily the point of contact. And then also, as we start to get ready to roll this out, we will then, particularly through Hopkins physicians as well as community oncology clinics, we will make it available for, your particularly in prostate or oncologists in breast or pancreatic cancer to be able to enroll as well. But we want to centralize our patient advocates because they're going to be our voice in the community, being able to explain that as to why this is important to our community.
Awesome. That's great. Thank you so much. And then Dr. Cooper asked a question about your mentoring philosophy. So she asked if you could please talk a little bit about your mentoring philosophy, and she acknowledged that your mentees have been incredibly successful.
Well, it's this sort of pull and tug. So one, give them freedom, but challenge them at the same time. so it's just it's a bit of challenge. I call it the directive mentoring. I meet regularly with them, with all of the trainees that we have in the lab. But I think our projects are big. I've created, you saw it's a tremendous amount of difficult techniques. I've had people in my lab that sort of concentrate on the actual technical part of it, because even being me a little bit more removed from the lab, I don't even get down to the newer techniques that we do. But when every person, when they meet with me, I'm challenging them mentally. I'm challenging their thought pattern. We're challenging their thought. We're encouraging them to not only follow my direction, but to lead. Even within the framework. So we have this framework disparity, but encouraging them to come up with their own ideas and following that. And sometimes allowing them to fail along the way is just as much as success, and having the freedom to grow. So by the time they leave me, they're confident individuals, and they know what it takes to fail. They know what it's like to succeed, and that they go out into the world and become confident individuals and scientists. And so that's kind of been my approach over the years. Been around long enough to stick around long enough to kind of refine that. And so that's been the approach that I've adopted.
That's great. I really loved hearing what you said, and shout out to Dr. Cooper, who is also an exceptional mentor. So I
Oh,
think
yes,
we'll have
absolutely.
a lot in common in that regard of, welcoming opportunities for growth, whether that looks like a failure, a temporary setback, or something that is successful. So
Right.
it's amazing. Are there any other questions from those who are in attendance? Any other thoughts?
I think folks are quiet. It was pretty impressive and a lot and for some people, attending today probably a little bit different from what we usually talk about, but I think it's really important for us to hear from our basic science colleagues, because we can identify, like Dr. Cruz said, these opportunities for collaboration. And a lot of times people are challenging us in our work to, sort of document, like, what's the link between these social and behavioral things you're measuring and doing, and what's the actual biology of disease. And we don't always have the specific markers we need to tell, or maybe by the time we've detected our diseases, they're at a stage where they're not as treatable as if we had markers that could detect some of the risk early on. So I found it fascinating and exciting to hear from you, and also hope that it'll similarly encourage other folks who are more on the population sciences side to think about building collaborations with colleagues in doing basic science of health disparities research.
Yes, I have, if you don't mind, I really must echo what Dr. Cooper just said. I loved this presentation. I'm not, as she's mentioned, we're not really accustomed to hearing a lot from the biological underpinnings of health disparities. And a lot of times, I will just speak for myself, we are always actually trying to say it's not just genetics, it is the social environment that is dictating, it's governing the patterns of these disparities. And I think it's invaluable to have this new research and just new insights into the interplay between the social conditions and the emergence of disease as a direct result of biology playing a role. And there was a point that you raised about some of the more recent work you're doing, I think around quadruple negative breast cancer, which
Yeah.
I didn't even know what that was a thing. But you mentioned something about the excitement of having research that really helps to bring about targeted treatment. So there's a drug and a companion diagnostic, and the way you worded it was matching the biology with the right therapeutic, and just the promise of that kind of thing for really providing the types of therapies needed for the groups that have been kind of systematically marginalized from the entire treatment experience, well, not just treatment, but even going back as far as screening. So it's just very exciting work.
Thank you. Thank you very much. And I do think, and I've been in those arguments, is it biology? Is it, what we think is both, and we think we're studying the same thing. It's just we're studying it differently. But I feel like we are studying the same thing. I think those behavioral actually are manifested, and that's why we see this biology. So happy to have some collaborations, because I'm showing you prostate and breast, but as I think caller one of said before, we see it's in colon, it's in kidney, it's in pancreatic. So it can't be just one. We're seeing it across, multi-cancer type in African American. So we absolutely think we're studying the same thing. We're just studying from different perspectives.
Yeah, absolutely. Well, thank you again so much for joining us, Dr. Yates. Oh, you know what? Never mind. I thought we were about to end, and then Kayla threw in an excellent question. So maybe we'll end with this question. What are your theories for why African Americans are not getting recommended screenings? Is it trust, bias, or neglect?
So here's the deal. The things that we found, when I was in Alabama, when approached, African American and Latino enroll more in clinical trials and clinical studies when approached correctly in a trusted provider. Okay, so that's one thing, and I think that's been replicated by many studies. So we know that exclusion is something that could be sort of systemic within the system and so forth. So I don't think it's trust per se, but I also, I was at Tuskegee, we had a bioethics center, and as being one of the leaders there, trust is to have trust, you have to be trustworthy. So that's sort of another issue. So we get into some soapboxes that we have. So trust is not going to be given blindly, and I think we have to do the work that suggests why a person should be enrolling in a screening or a clinical trial. So, for example, we're promoting that we need more diversity, but if I were an individual, I would want to see biology like I just showed to say why I need to be involved, instead of saying, hey, this thing was developed and now we want to test at the end when it takes years of development, and now we just want to think about diversity at sort of the sort of end stage, part when we get into the clinical setting. But we know it takes, tremendous amount of money and effort. It's just $1 billion to get a diagnostic to a clinic. So, all of that needs to sort of be incorporated. I think we got to revamp the system. And so that will create the trust, right? And make us trustworthy, that sort of break those barriers and provide the rationale why we need to enroll in a certain screen or a clinical trial in a certain population.
Wow. What a great way to end. And thank you so much, Kayla, for that excellent question. Just a quick reminder for the audience, if you could please complete the registration experience survey, that would be really great. As I mentioned before, we take all of the feedback to heart in terms of thinking about how to continue to improve upon our wonderful jam sessions. So thank you so very much, Dr. Yates. We're not only are we so honored to have had you join us today, but we're really looking forward to collaborating with you in the future and just cheering you and your team on and all the great work that is to come from all of these endeavors.
I look forward to working with you guys too, and thanks for having me.
Thank you.
It's been excellent. It's been very fun. Take care.